infectious diseases has caused the most preventable death around the world. Recent data from World Health Organization shows that among the top 10 causes of death in low-income countries, half of them are infectious diseases. Well, vaccines have helped wipe out a number of infectious diseases in human history, and vaccination remains the most cost-effective method for infectious disease control in the community, especially in the current era where antibiotics resistance become a huge problem. Vaccine development has progressed from using whole organism to the very precise peptide antigen. However, peptides themselves are usually poorly immunogenic and prone to degradation. This requires the expensive chemical coupling and the potentially toxic adjuvant. These things add extra cost and difficulty to the already existing challenging in immunization, such as the cold chain management from storage to transportation, and the high labor demand for highly trained personnel for performing the injection. Now, when talking about injection, I feel a shivering across the room. So our solution to break the wall of infectious diseases without the needle is a novel peptide delivery platform called Pilvex that utilizing the bacterial pilus structure to amplify, stabilize the peptide antigen and express it on the surface of Lactococcus lactis. Lactococcus is a food-grade bacterium that has the natural ability to boost immunity. And pili are long, thin, hair-like structure from the surface of many bacteria with the main function of attaching to the host cells. So the pilus are formed by the multiple copies of the backbone subunit that polymerize to form the pilus fiber. Pilvex, in a nutshell, is to engineer the peptide antigen within the backbone subunit, so the antigen can be protected in the rigid pilus structure and amplified through the pilus formation. And what we get is this, a bacterium that presents a large amount of antigen on the surfaces that can be given directly through the nose or the mouth. And this bacterium can be made into freeze-dried powder and stored and transport at room temperature, just like your probiotic pills. So in the pilot study, we immunized mice with the prototype Pilvex and showed that the immunization produced significant antibody responses in bloodstream and also the lung fluid and saliva. So these local antibodies can help guard the mucosal surface, which is the entry portal of many pathogens, and the circulating antibodies can help preventing disease from spreading. Thank you.
Thank you, Catherine. Any questions for Catherine?
Again, just a clarification really about the administration. That last picture you've got in the the Pilvex. The thing in the middle, is that still your bacterium, or what's the middle bit that carries all those
Yeah,
pili?
this is the Lactococcus bacterium that has been used for thousands of years in human society as the probiotic bacteria to fermented food. So it's a non-pathogenic bacteria that can easily be taken through as a form of nose spray or as solution or even as powder, just like your probiotic pills.
Cool.
So what is providing the adjuvant in this vaccine?
So the Lactococcus itself is a bacteria, and its cell surface has these sugar and protein compounds that being recognized by the human immune system. So the beauty of this is that we can even, we don't need to maintain this bacterium as live, because even the bacteria is dead or even heat inactivated, the surface of this structure still remains and is stable, and it still can stimulate immune responses.
Thank you. Are there any other questions for Catherine?
So is it easy to maintain accurate dosages?
Yeah, so this bacteria have been well established in the food industry and also as a bacteria factory for in the pharmaceutical industry. So the easiest way to quantify these bacteria is actually through the light. Like when they grow in the culture, it increase the turbidity. So using the light absorbance, you can measure how many bugs are in there. And these are like all established method for quantifying these bacteria.
Given the timing at the moment with measles outbreaks and the rise of anti-vax type sentiment, do you have any comment on how this new methodology of creating a vaccine might respond to some of the anti-vax sentiment that is out there?
Well, like currently, we are not identifying any new vaccine targets. So in our pipeline, our motto is to use established, like validated antigen to put into the system. And we have proved that with this system, it helps with delivery, it helps with stabilization. And the most important thing is that because of the formation of this vaccines, it reduced the cost significantly, and you don't need trained personnel to perform the injection. So that's really useful for the underprivileged settings where in fact effective vaccines are most needed. So as far as like as forming new vaccines, we are testing a few prototypes that use known antigen and put into our system. For example, the TB vaccine, the BCG vaccine, that's the only available vaccine for tuberculosis, but the protection rate is not reliable. And in our animal studies, it shows that it has comparable immunogenicity as the BCG vaccines, but it produces a safer type and more consistent type of immune responses that potentially means the superior efficacy than the existing vaccine.
But in terms of, it's just a follow-on question, in terms of reducing or limiting the need for an adjuvant, that would take care perhaps of some of the concerns?
Sorry, I didn't quite get the question.
With this new method, you
Yes.
no longer need an adjuvant in a
No.
vaccine, and that would possibly take care of some of the concerns that people have with vaccines?
Yeah, well, a lot of the concerns about vaccine is firstly the fear of needles, and then the chemical processing that can induce or introduce other impurities in the vaccine formulation, and also the adjuvant, because the adjuvant usually is derived from bacteria that can alarm the immune system. But with this vehicles is a food-grade bacterium that has known to pose no harm to the human body, but has the ability to enhance the immune responses.
That was a lot of questions. Good way to finish. Thank you, Catherine.